You walked out of a room, that's all. You crossed a threshold, maybe into a hallway, maybe into the kitchen, maybe out into the cooler air of somewhere less familiar, and something in you that you didn't ask to activate, activated. This happens constantly. You never catch it happening. Welcome to the Sleepy Biologist. Whoever kept this space going this week, through memberships, through super thanks, through something as quiet as leaving a comment, I'm glad you're here. This one's for the end of the day. There's a crease in the air when you move between rooms. Not literally. The physics of it is mundane, pressure equalizing, temperature shifting fractions of degrees, but the body doesn't experience it as mundane. The body experiences it as information. Sudden, usable, slightly urgent information about where it now is. The hippocampus, a curved, dense structure sitting deep in the brain, one on each side, is, among its many functions, a map maker. Not in a tidy way, not the way a GPS draws clean lines between points. It makes maps the way memory makes maps, imprecise at the edges, emotionally tinted, organized around what mattered and not around what was true. Inside this structure, individual neurons respond to location, not to light, not to sound specifically, not to any one feature of the environment, to place. A given neuron might fire reliably when you're standing at a specific spot in your kitchen and stay almost entirely quiet everywhere else. These are called place cells, and they were first identified in rats in the early 1970s. And for a while the discovery seemed so strange, so specific, so improbably precise, that the researchers who found them weren't immediately believed. What happens when you move from one room to another is not what most people would guess. The map doesn't just update, it switches. There's a word for it in the literature: remapping. When an animal or a person moves into a sufficiently different environment, the hippocampal place cells don't just shift their firing slightly. A largely different population of cells activates. The neurons that were encoding the previous room go quiet, and a new configuration lights up, drawing the new space from scratch, not building on what came before. Starting over. This is fast, faster than thought, faster than conscious recognition. You haven't decided the room is different yet. The hippocampus already wrote a new map. And here is the part that took me a while to sit with properly. The two maps don't blur together. They stay separate. One room, one map. Another room, another map, held in entirely different cellular patterns. The brain maintains these configurations in parallel, hundreds of them potentially, encoding every space you move through regularly, and switches between them with a speed that your conscious experience never catches up to. You don't feel the switch, you only feel the aftermath. That slight change in the quality of your attention when you step into a different space, the small, almost imperceptible shift that you might describe as a change of scene or dismiss as nothing, is the aftermath of remapping. A whole new population of neurons coming online, the previous population releasing its hold. And because these maps don't only encode location, because they encode context, expectation, associated emotion, the particular blend of sensory history that built the map in the first place, the switch isn't purely spatial, it's cognitive. Something about what you were doing in the last room, what you were feeling, what you were tracking, becomes less immediately accessible, not gone, less foregrounded. The load of the previous space partially stays there, not fully, not permanently. But in those first seconds after crossing a threshold, there is a genuine neurological gap, a brief interval where the old map is released and the new one is still assembling, a small cognitive clearing, a room of no rooms lasting maybe a second, maybe less. You probably don't notice it consciously, but you notice something. The thing is, and this is the part I find genuinely strange, the hippocampus isn't even the only structure doing this. The entorhinal cortex, which borders it and feeds information into it, runs its own spatial encoding. Grid cells there fire in geometric patterns across environments, creating a kind of coordinate system that gets reset with remapping. Head direction cells track orientation separately. Border cells fire when you're near the edge of a space. The brain is not taking a photograph of the room. It's running a full computational model of where you are, who you are in this space, what the space demands of you. And it's doing this constantly in real time, updating as you move, starting over when you cross from one context to another. All of this, just because you walked out of a room. There's a faint hum coming from somewhere in the building. Not a sound you'd name if asked, just a texture in the air, something the ears register below active attention. The new room has a slightly different version of it, or a different quiet. The nervous system clocks this in passing, files it, adds it to the new map. You didn't decide to notice it. The brain just did, because new environments get scanned differently than known ones. The filters are looser at first, more comes through. That's part of what feels like a breath of fresh air when you change your surroundings. Some of it is just this, a nervous system briefly letting more signal in before it decides what to start ignoring. And the room you just left? Still encoded, intact somewhere in the hippocampal patterns, waiting to be reinstated the moment you step back in. The map doesn't decay immediately. It holds, which means that on some level, cellular, architectural, subperceptual, you carry all the rooms you move through. Every threshold crossed adds a configuration that sits dormant in the tissue. The brain that falls asleep tonight is holding the geometry of every space you move through today, not consciously, not in any way you'd recognize as memory, but in the firing patterns of cells that spent all day learning where you were. What sleep does with all of that is a different question, but the maps are already made. Right now, without any instruction from you, a neuron is firing. Not because you told it to, not because you're thinking about anything in particular. It's firing because you are here, in this specific room, in this specific arrangement of your body relative to the walls and the dark and the particular quality of stillness that belongs to this hour. Move to another room, and that neuron will go quiet. It won't follow you. It has no interest in the hallway, in the kitchen, in the street outside. Its entire functional identity, if a neuron can be said to have such a thing, is organized around this place. There are others like it. Dozens, maybe more. Each one assigned to a location you inhabit with any regularity. Each one silent everywhere else. The cells are called place cells, and they live in the hippocampus, and the longer you sit with what they actually do, the stranger and more specific it becomes. John O'Keefe discovered them in 1971. He was recording electrical activity from individual neurons in the brains of freely moving rats, threading tiny electrodes through living tissue to catch the firing of single cells, and he noticed something that shouldn't have been surprising but was. Certain neurons spiked reliably when the rat was in a certain location. Not when it saw something, not when it heard something, not in response to any input O'Keefe could easily name, just here. The cell fired when the animal was here, and went almost entirely quiet when the animal was anywhere else. He called the regions of space that triggered each cell place fields. The cell wasn't responding to the room as a whole, it was responding to a specific zone within the room, sometimes small, a circle of maybe 30 centimeters, sometimes broader, but always defined, always its own. What O'Keefe had found, though it took years for the full weight of it to settle into neuroscience, was that the brain maintains a spatial map of the world, not as a stored image, not as a consciously accessible representation, but as a pattern of cellular activity that gets reinstated when you return to a known location. The map is the firing, the place is the pattern, and the pattern is specific enough that individual neurons can be assigned somewhat reliably to individual locations. The hippocampus you're carrying right now holds a large number of these patterns. Every room you return to regularly, your bedroom, your kitchen, a particular chair you sit in, has its own ensemble of place cells. Not always the same cells each time, and here is the part that tends to get quietly lost in popular explanations. The hippocampus doesn't perfectly repeat the same pattern every time you enter the same room. There's drift. Slight differences between one visit and the next, subtle shifts in which cells activate most strongly, as if the map is not a fixed recording, but something closer to a living habit, stable in its broad shape, variable in its fine detail, which means that the room you're in tonight is similar to, but not identical with, the hippocampal representation of this room from last night. Some cells overlap, some don't. The map from yesterday is still in there, somewhere, in whatever form memory takes in cellular tissue. Tonight's map is being written on top of it, or beside it, or into it. The relationship between them not fully understood, the exact mechanism of how old representations and new ones coexist still being worked out. I find this genuinely inconclusive in a way that feels appropriate. The brain is not a hard drive. The maps don't stack neatly. What does stack, what does seem to accrue over repeated visits, is something more like confidence. The more times you've been in a particular room, the more reliably the place cell ensemble activates, the faster the map assembles when you arrive, the more stable the representation becomes over time. Familiarity, at the cellular level, is partly this: an ensemble that fires consistently, that has been reinforced through repetition, that produces a spatial map with low variability. A room you've slept in for years has a different hippocampal representation than a hotel room you're in for the first time. Not better, not more detailed, just more consolidated, more certain of itself. The unfamiliar room requires more active construction, more input, more scanning. The place cells are still doing their job, still firing, still building a map, but the map is newer, less rehearsed, and the nervous system treats newer maps slightly differently. There's more sensory information being let through, more activity in the regions that handle novelty and uncertainty. And then there is the entorhinal cortex. If the hippocampus is where place cells live, the entorhinal cortex is the structure that feeds them, a region just adjacent, anatomically intimate with the hippocampus in a way that makes clean separation difficult. The entorhinal cortex contains a different kind of spatial cell entirely, grid cells, discovered by Edvard and Maybrit Moser in 2005, which don't respond to single locations but to a geometric pattern spread across the entire environment. A grid cell fires at multiple locations arranged in a precise triangular lattice, a repeating hexagonal pattern that tiles the space like a coordinate system. It doesn't care which room you're in, it cares about the geometry. The spacing between firing fields is fixed for a given cell. Different grid cells have different spacings, different orientations, and together they create something like a universal metric, a way of encoding not just where you are, but how far you've travelled, in what direction, at what scale. The place cells tell the brain, here. The grid cells tell the brain, how far from there to here. Between them, and with the help of head direction cells and border cells and several other specialised populations, the brain builds an internal model of space that runs continuously, updates constantly, and never asks for your participation. There's something that stands out to me about all of this, something I keep returning to without being entirely able to name it, which is the sheer commitment of it. A neuron that fires only here, only in this specific zone of this specific room, and waits silently everywhere else. The waiting is most of what it does. Most of its existence in terms of time is not firing, is being still, is holding a pattern in readiness for the moment you return to the location it was assigned to, and it will still be there. You could leave this room for 6 months and come back and the ensemble would reassemble, not instantly, not perfectly, perhaps with more drift than yesterday's version, but the cells would recognize the context and the map would reconstitute itself. Something of it would still be there. The tissue holds it. This is part of what makes certain rooms feel, on return, so immediately and specifically themselves. Not just familiarity in the vague sense, something cellular, something faster than recognition, the place cell ensemble activating before the conscious mind has finished registering that it's back. Somewhere it knows, the body arriving before the thought does. You felt this, probably in a childhood home, or a room you lived in for long enough that it settled into something like the body's own geometry. The quality of the familiarity is different from remembering. It's spatial before it's anything else. It doesn't require thought to produce it. That's because it isn't thought. It's the pattern reinstating. The old ensemble coming back online, matching the geometry of the space, assembling the map it built over years of repetition into something coherent again. The room hasn't changed. The cells haven't forgotten. The hippocampus is working right now, not hard probably in a room this familiar, running the ensemble for this space, maintaining the map, cross-referencing position with the entorhinal grid, doing quietly what it has always done in every room you've ever stayed in long enough to sleep. You carry all of them, every room that mattered, encoded to some degree in the tissue, stored not as image or language, but as pattern, as readiness, as a cellular posture that reassembles when you return to the right coordinates. The room you sleep in has one of the most consolidated maps your hippocampus holds. It's been rehearsed every night. You didn't notice the hum until just now. Maybe you still don't. Maybe reading that sentence made you listen for something and you found, what exactly? A faint electrical drone from somewhere in the walls. The particular silence of a room that isn't actually silent. The sound of your own breathing, which you'd stopped counting as sound. You weren't ignoring these things. That's the part worth slowing down for. The nervous system does something that has a name, habituation, and the name makes it sound deliberate, like a choice the brain makes after consideration. It isn't. It's faster than choice, older than choice, and it operates well below any level of processing that could be called conscious. Here is what happens roughly. A stimulus arrives. The sensory systems register it. The brain checks. Is this new? Is this relevant? Is this something that requires a response? In the early moments of a repeated, unchanging stimulus, a hum, a background vibration, the distant sound of traffic, the answer is uncertain enough that the signal gets passed upward, gets attended to, gets processed. But if nothing changes, if the hum stays the same pitch, the same volume arrives with the same regularity, the brain begins to update its prediction. This signal is constant. It has not yet produced a consequence. It is not so far a threat or an opportunity or anything requiring action. And gradually over minutes or hours, depending on the stimulus, the neural response to it diminishes. Not because the sound stops, because the brain stops bothering to respond to it. The technical term is neural adaptation, and it happens at multiple levels simultaneously. Sensory receptor cells can reduce their firing rate in response to a sustained, unchanging input. The receptor itself becomes less responsive, not just the downstream processing. Further up the chain in the cortex, the representation of the stimulus shrinks. The neurons that were active in response to it gradually quiet. The sound is still arriving at the ear. The cochlea is still converting it to electrical signal. The auditory nerve is still carrying that signal toward the brain. But somewhere in the processing, the gate has been narrowed. The signal is getting through in reduced form, or being actively suppressed. Or, and this is the part that's still not fully settled in the research, being predicted so accurately that the brain simply generates the expectation of the sound and stops needing the sound itself to confirm it. The hum becomes something the brain already knows will be there. And what the brain already knows stops being interesting. This is not a flaw. It would be worth saying that once, clearly, before moving on. Habituation is not the nervous system failing to pay attention. It is the nervous system making a very efficient decision about where attention is worth spending. A brain that responded with full alertness to every unchanging background stimulus would be a brain that couldn't function, overwhelmed by the constancy of the world, unable to detect what actually changed. The whole architecture depends on filtering. What you hear right now is not the full acoustic environment of this room. It's a curated version. The things the brain has decided haven't been stable long enough to safely ignore. The things that changed recently. The things that don't yet have a confirmed prediction attached to them. The rest has been subtracted. You're living inside an edited version of the room's noise. What happens in a new environment, and this connects quietly to everything the previous chapter was building toward, is that the habituation is briefly undone. Not completely, not permanently, but a new room carries stimuli that don't yet have predictions attached to them. The hum of this different building, the creak of a different floor, the texture of a different silence. The brain doesn't have a filed expectation for any of it yet. So it responds. The neural representation is fuller, louder in a sense, even if the actual sound volume is lower. The new room is neurologically noisier than a familiar one, even if it's quieter in every measurable way. This is part of why sleep is harder in an unfamiliar place. It has a name in sleep research and the first night effect, and one of its components is exactly this. The auditory system in an unfamiliar environment has not yet habituated to the room's background sounds. The sounds are being processed more fully. The brain is still deciding what they mean. There's a low-level alertness, not fear, not anxiety necessarily, just the nervous system holding a slightly higher readiness for sound-based information that makes the transition into deep sleep slower, shallower, more fragmented. One hemisphere, some research suggests, stays slightly more active than the other on that first unfamiliar night, a kind of divided vigilance, part of the brain trying to sleep, part of it still listening for the sounds it doesn't have predictions for yet. The room you sleep in regularly doesn't do this. It's been habituated to. Its sounds are predicted before they arrive. The brain has, over many nights, filed away the particular acoustic fingerprint of this space, the pipes, the ventilation, the quality of the quiet, and flagged it as known, safe, not requiring response. So the sounds don't wake you. Mostly. There's a wrinkle that's worth mentioning, though, because it's one of the stranger edges of this process. The sudden absence of a habituated sound can wake you. If the refrigerator stops humming, the one you stopped noticing months ago, there is sometimes a brief moment of alertness. Not because a new sound arrived, but because a predicted sound didn't. The brain was generating the expectation of the hum, and the hum wasn't there to confirm it. That mismatch, prediction without confirmation, is its own kind of signal. A small specific jolt, silence, in the wrong place is louder than noise. Right now, the sounds in this room are probably somewhere in the middle of that process for you, some of them fully habituated, old sounds predicted, filed, some of them newer, still being assessed. Maybe something shifted recently, a change in weather, a different kind of quiet than last night's, some small acoustic difference the brain is still deciding whether to care about. It's checking. It will probably decide not to, and then it will stop hearing it. The way it stops hearing everything, eventually that doesn't change. There is a structure in the brainstem so small that if you held a human brain in your hands and knew exactly where to look, you still might not find it without magnification. A few millimeters across, a dense cluster of neurons tucked near the floor of the fourth ventricle, in a region called the pons, stained slightly blue in preserved tissue by a pigment the cells produce called neuromelanin. Its name is the locus coeruleus, Latin for blue place. And its axons, the long projecting fibers that carry its signals outward, reach into nearly every region of the brain. The cortex receives them, the hippocampus receives them, the cerebellum, the spinal cord, the amygdala, the thalamus. All of them receive projections from this one small cluster of perhaps 50,000 neurons in a structure that weighs, in total, less than a grape. The numbers are slightly different across individuals and across species and across sources, and the exact count is less important than the topology. One source, many destinations. A single origin point for a signal that arrives almost everywhere. That signal is norepinephrine. And one of the most reliable things that triggers its release, not the only thing, not the most dramatic thing, but one of the most consistent, is novelty. Not danger, not pain, not the kind of arousal that comes with threat. This distinction matters, and it's one that tends to get collapsed in popular explanations of the norepinephrine system, which often leads with stress and adrenaline and the body preparing for something difficult. That version is real. Norepinephrine does rise sharply under threat, does prepare the body for action, does play a role in the fight or flight cascade. But that is one end of a wide dynamic range, a spectrum with alarm at one extreme and something much quieter at the other. The quieter end is what happens in a new hallway. What happens when the environment changes and the change is not threatening but is genuinely new. New enough that the nervous system doesn't yet have reliable predictions for it. The locus coeruleus responds to this too, not with the sharp spike of threat detection, with something smaller, a modest elevation in firing rate, a slight increase in the amount of norepinephrine being released into the terminal fields across the brain. A lamp turned slightly higher in a room you thought was already bright. What norepinephrine does at this moderate level is adjust the signal-to-noise ratio of neural processing. The phrase is borrowed from engineering and doesn't map perfectly onto biology, but the basic logic holds. In neural circuits, there is always some baseline level of activity, spontaneous firing, background noise, the ongoing hum of cells doing their resting business. A signal arriving into this background has to be detectable above it. Norepinephrine, at moderate levels, has the effect of suppressing some of that background activity while simultaneously increasing the responsiveness of neurons to incoming stimuli. The baseline noise drops slightly. The response to meaningful input rises slightly. The result across the brain is something like increased acuity, not the frantic hypervigilance of high norepinephrine release, which eventually degrades processing rather than improving it. Something more measured, an environment being scanned with slightly sharper instruments than the ones that were running a few minutes ago, in the familiar room, where everything was already predicted and the locus coeruleus had little reason to adjust. The hippocampus, receiving this norepinephrine signal, responds in ways that seem to affect how spatial information is encoded. There is evidence, not entirely settled but accumulating, that norepinephrine release during novelty exposure enhances the formation of new place cell representations. The new map, built when you walk into a new space, may be encoded more robustly when norepinephrine is present than when it isn't. The brain sharpens its map-making precisely when the map is newest. There is something almost too tidy about this that makes me want to be careful not to overstate it. Biology rarely works in such clean functional stories, but the correlation exists, and the mechanism has enough support that it's worth sitting with. The same signal that increases perceptual acuity also seems to strengthen the encoding of new spatial context. The novelty response isn't just attention, it might be a form of preparation for memory. The prefrontal cortex receives norepinephrine too, and its response there is different, governed by different receptor subtypes with different sensitivities to the molecule. At moderate concentrations, the kind produced by mild novelty, the kind produced by a new hallway rather than a dangerous one, norepinephrine acting on certain receptors in the prefrontal cortex tends to improve what researchers call working memory. The capacity to hold information active, to keep track of what's currently relevant, to maintain a representation of the current situation while operating within it. Walking into an unfamiliar environment and being able to orient yourself, tracking the exit, building a mental layout, keeping multiple spatial relationships in mind simultaneously, draws on this, and the norepinephrine that arrived because the environment was new may be part of what supports it. The molecule that responded to novelty also helps you navigate novelty, not designed this way presumably, just selected for, over a very long time, in animals that needed to survive unfamiliar terrain. What fades is important too. The locus coeruleus doesn't sustain elevated firing indefinitely in response to a stable novelty. If the new environment remains stable, if the new hallway doesn't keep producing surprises, the firing rate begins to return toward baseline. The norepinephrine signal diminishes. The slight sharpening that came with it diminishes with it. This is habituation at the neuromodulatory level, running in parallel with the sensory habituation from the previous chapter. The brain not only stops hearing the new room sounds, it also gradually withdraws the neurochemical amplification it brought to the task of hearing them. The room becomes ordinary. The locus coeruleus quiets. The signal-to-noise ratio settles back toward its resting state. By the time a room is fully familiar, slept in, habituated to, mapped completely, the locus coeruleus barely stirs when you enter it. There's nothing new to report. The predictions are all in place. The map exists and is simply reinstated rather than built. The norepinephrine that flooded gently through the cortex on the first night in a new space, or the first time you walked into a room that mattered, has long since stopped arriving in those quantities. What remains is the map it helped build. The encoding it may have strengthened. A room that the brain holds with a certain confidence now, cellular and quiet, because once, when it was new, the chemistry was right for making it permanent. There's a kind of tiredness that isn't sleepiness. You felt it. The day wasn't physically hard, maybe. You sat mostly, moved between rooms, looked at screens, read things, replied to things, and yet by evening there is something depleted that sleep alone doesn't fully explain. Particular flatness in the quality of thought, a resistance to starting anything new, the sensation, if you tried to name it, of a muscle that has been slightly tense for a very long time. That muscle is real, it's just not in your body. The prefrontal cortex, the region most associated with planning, with sustained focus, with the kind of thinking that requires holding multiple things in mind at once, is metabolically expensive to run at full capacity, not dramatically so, not in the way a sprint is expensive, but steadily across hours, in a way that accumulates without announcing itself. Directed attention is the term researchers use. The deliberate, effortful kind would sample. The attention required to stay focused on something that doesn't inherently compel you, to filter out distraction, to maintain a task representation against the pull of everything else that wants processing. This is what you use when you're reading something dense, or working through a problem that resists easy solutions, or sitting in a meeting that requires you to track multiple threads simultaneously. It works by inhibition. That's the part that tends to get left out of casual descriptions of focus. Paying attention to one thing requires the active suppression of everything else, a constant neural effort to quiet the signals competing for processing. The focus is only partly about amplifying the target. It's equally about dampening everything that isn't the target. And dampening takes energy. By late afternoon, for most people, something in the quality of this changes. The inhibitory effort becomes less efficient. Distractions that would have been smoothly filtered earlier in the day start getting through. The task representation becomes less stable. The mind drifts more easily, returns more slowly. Decisions that felt clean in the morning start to feel effortful in a way that isn't about their actual difficulty. This is attentional fatigue, and it has a biology that goes beyond the brain is tired. There are specific deficits that emerge. Inhibitory control degrades before other cognitive functions, meaning the filtering mechanism fails before the raw processing does. You can still think, you just can't as easily control what you're thinking about. The posture of directed attention becomes harder to hold. Stephen Kaplan, a psychologist who spent decades studying the relationship between environments and attention, proposed a framework in the 1980s that remains useful even where it remains contested. He called it attention restoration theory, and its central claim is this: directed attention is a finite resource that depletes with use, and certain environments, specifically natural ones, contain features that allow it to recover. Not by providing rest in the sleep sense, by engaging a different kind of attention entirely. Kaplan called it involuntary attention. The distinction matters. Directed attention requires effort to maintain. Involuntary attention requires none. It is captured by the environment rather than deployed toward it. The movement of leaves, peripheral ripple of water, the unpredictable but non-threatening complexity of a natural landscape where there is always something shifting at the edge of vision without any of it demanding response. These things hold the attention without asking anything of it. And while involuntary attention is running, while the environment is doing the work of engagement, directed attention, the effortful kind, gets something it rarely gets during a normal day: a chance to stop. The biology underneath this is still being mapped with some precision, and I want to be careful here not to make it tidier than it is. What the research does show, with reasonable consistency, is that exposure to natural environments, or even to images of natural environments, though less robustly, produces measurable changes in physiological markers associated with stress and arousal. Heart rate variability improves, cortisol levels drop slightly, prefrontal activity, as measured by certain imaging protocols, shifts in ways consistent with reduced effortful processing. The environment does something. What exactly it does, and through exactly which mechanisms, is still being worked out. What seems most likely is not a single pathway but several converging ones. The reduced demand for inhibitory effort, the parasympathetic shift that comes from environments perceived as safe and non-threatening, the norepinephrine dynamics from the previous chapter running in reverse, a familiar low-novelty environment requiring less neural amplification, allowing the system to settle. And something else, harder to quantify. The particular quality of attention that natural complexity produces is not quite wakefulness and not quite drowsiness. It's something between them. Engaged but undemanding, curious but unurgent. The gaze moves without intention. Sound arrives and is registered without being tracked. There's a softness to the processing, a looseness in the filtering that directed attention never allows. You probably know what this feels like without having a name for it. The particular quality of sitting somewhere with moving water nearby or watching something in the peripheral distance. Trees, clouds, the ordinary drift of an outdoor space. And noticing after a while that you haven't been thinking about anything in particular. Not because you fell asleep, because the environment was doing enough that your brain could stop doing the rest. That's not nothing. That's the directed attention system going briefly offline. The room you're in right now has a relationship with your attention that's been building all day. If it's familiar, if this is a room you associate with rest, with the end of effort, with permission to stop, then something about being here is already different from being in the room where the work happened. The association isn't just psychological. Familiar low-demand environments reduce the drive toward task maintenance. The prefrontal cortex, which has been running inhibitory effort since morning, receives fewer signals demanding its engagement. The posture of attention starts to release, not because you decided to let it, because the room told it something. Directed attention doesn't switch off cleanly, it loosens gradually, the way a grip does when the thing being held is set down, not instantly, but in stages, the tension releasing in layers over minutes rather than seconds. By the time you're horizontal in the dark in a room the brain has filed as safe and familiar and finished with the day, most of what has been held tightly all day is somewhere in the process of letting go. The effortful posture, the one you've been holding since morning without knowing you were holding it, is finally without a task to justify it. It's looking for somewhere to put the weight down, and the familiar dark and the particular silence of this room and the body that horizontal in the way that signals to the nervous system that the directed part of the day is over. All of it is the surface it's been looking for. Memory is not stored the way most people imagine it is stored. It is not filed, it is not archived in some clean internal library, where retrieval means locating the right drawer and pulling out an intact document. The document metaphor, tidy, retrievable, stable, is almost entirely wrong. And the wrongness of it matters for understanding something that happens every time you move between rooms, every time the environment around you changes enough that the brain registers a genuine shift in context. What actually happens is closer to this. A memory is encoded not as a fixed representation, but as a pattern of neural activity that was assembled in a particular moment, in a particular context, with a particular set of conditions surrounding it. The sensory texture of the space, the internal state of the body, the preceding thoughts, the hormonal climate of the hour. All of that becomes part of the encoding, not just what was remembered, but the whole surrounding circumstance of the remembering. And retrieval later is not a clean read out. It is a reconstruction, an attempt to reassemble the original pattern using whatever cues are currently available. The more closely the current conditions match the original encoding conditions, the more completely the pattern reassembles. The less closely they match, the more the reconstruction struggles. Context isn't the background to memory. it is part of the memory itself. This principle has been studied rigorously since the 1970s in experiments that still feel, when you read them carefully, mildly strange in how clearly they demonstrate something the intuition resists. Godden and Baddeley in 1975 tested divers who learned word lists either underwater or on land, then were tested for recall in the same or different environment. Words learned underwater were recalled better underwater. Words learned on land were recalled better on land. The environment at encoding and the environment at retrieval shared something, some set of contextual cues that either supported or disrupted the reconstruction of the memory. The finding has been replicated in different forms with different variables across decades. Internal state matters too. Memories encoded while calm are retrieved more easily while calm. Memories encoded under stress carry the stress as part of their structure. Mood, arousal level, even specific smells present during encoding. All of them become threaded into the pattern, and their absence during retrieval creates a small but real gap between the stored pattern and the current conditions. The gap is the forgetting, not the loss of information necessarily. The loss of the conditions that make the pattern fully reassemblable. So what happens to memory when the context changes, when you walk out of one room and into another? Something does. The research is clear enough about this, even where its mechanisms are still debated. Context shift produces a reduction in the accessibility of memories formed in the previous context. Not erasure, not deep forgetting. A reduction in how readily they surface, how fully they reconstruct, how easily they're accessed without deliberate retrieval effort. The memories are still encoded somewhere in the tissue. They're just harder to reach from here. There is a structure in the hippocampus, a subregion called CA3, dense with recurrent connections, capable of pattern completion from partial cues, that seems particularly involved in this process. CA3 can reconstruct a full memory from a fragment of the original encoding conditions. Smell a particular thing, and suddenly a whole afternoon reassembles. Hear a sound from the right time period, and a whole texture of experience becomes briefly accessible. But pattern completion depends on the fragment being similar enough to the original that the reconstruction can get started. A context shift changes enough of the available cues that CA3's pattern completion works less smoothly. The fragment offered by the new context doesn't closely match the encoding conditions of the previous room's memories. The reconstruction stalls at the beginning. What the brain tends to surface instead are memories encoded in context similar to the current one. You walk into the kitchen and find yourself thinking about something that happened in a kitchen. You walk into a room associated with the evening and find the accessible memories tilting toward other evenings. The current context is pulling certain patterns toward the surface and leaving others at depth. This is not random. It is context-cued retrieval operating exactly as it was built to operate, prioritizing what is likely to be relevant here in this context over what was relevant in the previous one. The part I find most worth sitting with is what this means for the weight carried between rooms. If memory is context-dependent, then so is the cognitive and emotional texture that rides alongside memory. The particular quality of unfinished thinking that was running in the previous room, the half-formed worry, the unresolved task representation, the low-grade emotional residue of an earlier conversation, those states were encoded in a context. They have a retrieval address that includes the room they were running in. Move context sufficiently, and their accessibility changes. Not because they've been resolved, not because you chose to set them down, because the pattern that holds them requires conditions that are no longer present. The new room doesn't have the retrieval cues. The reconstruction doesn't fully initiate. The weight that felt so immediate in the previous context becomes not absent, but harder to locate, less sharp at the edges, somewhere behind you rather than in front. This is part of why changing your environment works when it works. Not because the problem's dissolved, because the context that held them is no longer the active context. You stepped out of the retrieval conditions. The hippocampus is doing this constantly, invisibly, as you move through your day, sorting what's accessible by what's contextually relevant, keeping the current context memories foregrounded while the others settle below the surface. It is not protecting you from anything. It is not managing your emotional state in any intentional sense. It is simply doing what context-dependent memory systems do, organizing retrieval by relevance, where relevance is defined, in large part, by where you are. The context is the filter. The room is part of what decides what you can easily think. What happens to the memories that become less accessible after a context shift is a question I don't think has a fully clean answer. They don't disappear. The evidence for that is strong enough to say plainly. Deep forgetting, the actual loss of encoded information, is a slow process. Occurring over very different timescales than a walk between rooms. What changes is access, not existence. But whether they are reconsolidated in some form during the context shift, whether the reduced accessibility itself changes them in some way, whether repeated context shifts, a life lived across many rooms, many environments, many contextual frames, shapes the overall architecture of what remains easily retrievable over years, that part is genuinely less settled. The hippocampus keeps its own accounting. What it discards, and what it keeps near the surface, and what sits waiting at depth for the right conditions to bring it back, the full logic of that is still being worked out, one experiment at a time, in laboratories where the questions keep turning out to be harder than the previous question, which feels somehow right. You breathed in when you entered the room. You always do. It's not a choice. The respiratory cycle doesn't pause at thresholds, doesn't wait for permission, but the breath that happens when you first cross into a new space carries something the subsequent breaths don't. The olfactory system gets its first unhabituated read of the air. Everything the room is putting into the atmosphere, temperature, humidity, volatile compounds from the materials and surfaces, and the particular biological history of the space, arrives at the olfactory epithelium in that first inhale without prediction attached to it. A few seconds later, adaptation begins. By the time you've been in the room for 2 minutes, most of what you smelled first is already fading. Not because the molecules have left the air, because the receptors are already adjusting. The olfactory epithelium sits high in the nasal cavity, a small patch of specialized tissue, a few centimeters square, covered in receptor neurons that extend hair-like cilia into the mucus layer, where airborne molecules dissolve and bind. There are roughly 400 different types of olfactory receptor in humans, fewer than many other mammals, more than is sometimes assumed, and each receptor type responds to a different molecular shape, or more precisely, to a different set of molecular features. A given odor molecule might activate several receptor types simultaneously, and the pattern of activation across the receptor population is what the brain eventually reads as a specific smell. The signal travels from these receptor neurons directly to the olfactory bulb. And this is where the anatomy becomes unusual. Almost every other sensory system in the brain routes its signals through the thalamus first. The thalamus sits near the center of the brain, receives incoming sensory information, and relays it to the appropriate cortical areas for conscious processing. Vision goes through it, hearing goes through it, touch goes through it. The thalamus is, in a rough sense, the sensory relay station, the place where raw signals get directed before the cortex makes something of them. Olfaction doesn't go through it. The olfactory bulb projects directly, without thalamic relay, into the piriform cortex, the amygdala, and the entorhinal cortex. The amygdala, which is centrally involved in emotional processing and emotional memory. The entorhinal cortex, which feeds directly into the hippocampus. These connections are not detours or secondary pathways. They are the primary route. Smell reaches emotional and memory structures before it reaches the parts of the brain that produce conscious awareness of smelling. This is why certain smells do what they do before you've decided to let them. The particular smell of a childhood home, paint or wood or something you couldn't name if asked, arrives in the amygdala and entorhinal cortex before it arrives in any structure capable of recognizing it as a memory. The emotional texture of the memory can begin assembling before the conscious mind has identified what it's smelling. You feel something first. You know what it is second. Sometimes you never know what it is. It's not mystical, it's just anatomy. The pathway is shorter. What a room's air carries is more varied than it might seem. Volatile organic compounds off-gassing from furniture, flooring, paint. Carbon dioxide levels that shift with how many people have been in the space and how recently. Humidity that changes the density and feel of the air in ways the respiratory system registers even before smell does. Microbial communities, the particular bacterial ecology of a space, contributing their own volatile signatures to the smell. And then there are the human chemical signals, though the science here is less settled than popular accounts tend to suggest, and I want to be careful not to overstate it. The evidence for functional human pheromones, airborne chemical signals that produce specific behavioral or physiological responses in other people, is considerably weaker than the evidence in other mammals. There are candidate molecules, there are studies with intriguing results, but the clean story of human chemical communication through smell is not yet a clean story, and anyone presenting it as one is moving faster than the data. What is clear is that the olfactory system is sensitive to far more than it consciously reports. Most of what the nose detects never reaches conscious perception. It goes somewhere else first. The new room's smell matters, then in ways that operate below the level of noticing. If the air carries familiar molecules, the particular signature of a space you've returned to many times, the amygdala and hippocampus receive a context signal that arrives before the eyes have finished adjusting, before the place cells have fully assembled the spatial map. A chemical confirmation. This is known. This has been here before. The emotional associations of this space are accessible. If the air is unfamiliar, that confirmation doesn't come. The olfactory input adds to the novelty signal rather than dampening it. One more dimension of the environment that hasn't been habituated to. One more set of molecules without a filed prediction. The room you sleep in has a smell you stopped noticing, probably years ago, if you've been sleeping there long enough. The olfactory adaptation happened gradually. The receptor response diminished with repeated exposure, and now the molecules that constitute the particular atmosphere of this room arrive at the epithelium and produce almost nothing. No signal worth forwarding, no pattern worth assembling, no emotional content worth surfacing. But the pathway is still there. The amygdala is still receiving. And at some level below conscious smell, below anything you'd call noticing, the air of this familiar room is still arriving where it always arrives. Still confirming what it always confirms, that this is the room, that you've been here before, that whatever this air has always meant to the structures that receive it first, it still means that now. There's a weight that arrives so gradually you stop feeling it as weight. It starts as something you'd name, a difficult week, a sustained pressure, something the body flags as different from ordinary. The nervous system responds appropriately. Cortisol rises. The sympathetic branch of the autonomic nervous system nudges the baseline slightly upward. Heart rate a fraction higher, breathing a fraction shallower, the muscles holding a low, persistent readiness that doesn't quite release between demands. This is the system doing what it was built to do. The problem isn't the response. The problem is what happens when the thing that triggered the response doesn't resolve, when the week becomes a month, when the pressure becomes the texture of the days themselves, rather than a discrete event sitting inside them. Cortisol is released from the adrenal cortex, small glands perched above the kidneys, in response to signals that originate in the hypothalamus, travel to the pituitary, and then arrive at the adrenal glands as a cascade of hormonal instruction. In acute stress, the release is sharp and purposeful. The body mobilizes glucose, sharpens attention, suppresses processes that aren't immediately necessary. It is, in its original context, an elegant and efficient response. What it is not built for is continuity. The same elevation that serves the body well over minutes or hours begins to do different things over weeks. Chronically elevated cortisol shifts the immune system's behavior, not simply suppressing it, as the shorthand goes, but altering its priorities in ways that tend toward low-grade systemic inflammation. It affects the hippocampus, which carries high densities of cortisol receptors in ways that researchers are still mapping, but that seem to involve changes in the ease with which new memories form and old ones are retrieved. It changes sleep architecture, not eliminating sleep, but reducing the proportion of slow-wave sleep, the deep restorative phase, in ways that compound over time. None of this feels like what it is. It feels like you're tired but can't rest fully. You're not ill but not quite well. You're managing, but the managing itself is heavy in some way that resists description. The sympathetic nervous system, running at a slightly elevated baseline, produces something that becomes so constant the body stops registering it as elevation. This is the strange biology of adaptation. The same mechanism that quiets a background sound eventually quiets a background state. The mild tachycardia, the slightly elevated blood pressure, the faint persistent muscular tension in the shoulders and jaw, and the muscles along the back of the neck. These things, if they arrived suddenly, would be immediately noticeable. Introduced gradually, maintained confidently, they become invisible. The body recalibrates its sense of normal. And once it has, there is no longer an internal alarm to signal that something is different from how it should be. You are simply this, now. This is simply how you are. The reveal, if it were, usually comes from the outside, not from introspection, which is already adjusted to the new baseline, not from a decision to check in with the body, because the recalibrated body doesn't know what it would be checking against. The reveal tends to come from contrast. From the moment a sustained environmental stressor is removed, or from the moment the body is placed in an environment sufficiently different from the one that has been maintaining the load. A vacation that takes 3 days to feel like anything. A weekend that doesn't deliver relief until Sunday afternoon, when something finally releases and you realize, only in the releasing, how tightly it was held. A walk that achieves nothing particular but during which the jaw unclenches by itself, uninstructed, because the environment stopped requiring the clench. The contrast is the only diagnostic available. You find out you are carrying it by putting it down, or by stepping somewhere the carrying doesn't follow as easily. This is what environment change does that almost nothing else does with the same speed. The load that has become invisible, the cortisol-elevated, sympathetic humming, muscular tension-sustained chronic state that the conscious mind has entirely stopped flagging as unusual, doesn't have a retrieval address in the new environment. Recall what the previous chapter covered: context-dependent memory, context-dependent state. The cognitive and emotional texture of the previous environment is encoded in that environment. Move the context sufficiently and the accessibility of that texture changes. The body doesn't know it's carrying a load, but it knows in some sub-perceptual way that the new place doesn't feel like the place where the load lives. Something loosens, not because the situation that produced the stress has changed, not because anything has been resolved or decided or set down consciously, because the nervous system, ancient and efficient, is running a context check, and the current context doesn't match the context where the alarm was calibrated. The alarm, not finding its triggers quiet slightly. Cortisol follows a diurnal pattern under ordinary circumstances, high in the early morning, declining across the day, reaching its lowest point in the hours around midnight. Chronic stress flattens this curve, reducing the amplitude of the morning peak and elevating the evening trough. The daily rhythm of the hormone becomes less rhythmic. Sleep, which depends partly on that evening cortisol trough for its initiation, becomes harder when the trough is elevated. Not impossible. The sleep pressure built by adenosine accumulation during the day is eventually powerful enough to override most interference, but different. Lighter, less restorative. A sleep that doesn't quite reach the depths it should. This is part of why the chronic load compounds itself. The biology that would best recover from sustained stress, slow-wave sleep, the glymphatic clearing, the memory consolidation and emotional processing that deep sleep provides, is the biology most disrupted by the sustained stress that requires it. The body trying to recover the resource it needs to recover, finding the resource slightly depleted. The room you're in now has a cortisol signature. Every environment does, not literally, not in the air, but in the body that has spent time there. The rooms where the load was heaviest have a physiological familiarity to them that the nervous system reads, on re-entry, as a prompt to reinstate the associated state. The rooms where the load was absent have a different signature. The rooms where the body learned to release, where the sympathetic hum quieted, where the jaw unclenched on its own, where the cortisol curve was allowed to follow its natural descent. Those rooms carry a kind of physiological permission. This room, if you've been sleeping here long enough, may be one of them. The nervous system recognizes it, not consciously, not as a thought, as a slight shift in the baseline, a small release in the muscles of the neck and shoulders that you might not notice unless you are already noticing. A breath that goes slightly deeper than the one before it, unremarkable, uncelebrated, the body beginning to remember that here, in this particular dark, in this particular familiar quiet, the load doesn't have to be carried at the same height. It can be set down a little. Not all of it. Not tonight. But some of it. Enough. For a long time, neuroscientists looking at brain imaging data noticed something they didn't have a good explanation for. When a person was given a task, asked to solve a problem, read something carefully, focus on a target, certain brain regions reliably activated. This was expected. But a separate set of regions reliably deactivated. Went quieter when the task started, came back online when the task ended. These regions weren't doing nothing when the task was running. They were being suppressed, held down, actively inhibited by the task-positive networks doing the focused work. When the task stopped, they returned. The network formed by these regions has a name now, the default mode network, sometimes called the DMN, though the name is a little misleading, suggesting a kind of factory setting, a placeholder state, something the brain does while waiting for something more important. That framing turned out to be wrong, or at least insufficient. The DMN isn't idling, it's running something. The question of exactly what it's running is still being argued over in ways that feel genuinely productive rather than merely unresolved. What researchers have found across years of imaging studies is that the DMN tends to be active during certain specific mental states. Self-referential thinking, thoughts about yourself, your past, your future, your relationships. Mental simulation, imagining scenarios that haven't happened. Perspective taking, considering what someone else might be thinking or feeling. Memory retrieval that isn't cued by an immediate task. It's the network that runs when the mind is, loosely speaking, with itself. Not empty, with itself. During a task-heavy day, the DMN spends a lot of time suppressed. Every time directed attention is deployed, and every meeting, every document, every sustained period of focused effort, the task-positive networks activate and the DMN is held quiet, not completely silent. There's evidence that it continues to run at some level, even during focused tasks, particularly during the brief pauses between demands. But its full expression is constrained by the ongoing requirement to direct attention outward, toward the task, away from whatever the DMN would otherwise be doing. This suppression isn't neutral. The DMN seems to push back against it in some sense. Mind-wandering during tasks is partly the DMN re-emerging despite suppression. Thoughts about self and future and unfinished personal concerns breaking through the focus in the moments when the inhibitory pressure relaxes slightly. You know this feeling. The unbidden thought in the middle of reading something. The sudden arrival of an unrelated concern during a conversation. Not a failure of willpower, a network reasserting itself during a momentary gap in suppression. What happens in an unfamiliar environment has an interesting relationship with all of this. A familiar room, the office, a regular workspace, a space strongly associated with a specific type of task, carries contextual cues that prime the task-positive networks. The environment itself is a kind of instruction, nudging the brain toward the attentional posture the space is associated with. This is the context dependence from earlier chapters running through the attentional system rather than the memory system, the room signalling what kind of processing is expected here. An unfamiliar environment doesn't carry those cues, or carries different ones. Cues associated with exploration, with soft environmental attention, with the kind of present moment sensory engagement that doesn't require the sustained directed focus of task performance. There's nothing in the new space telling the prefrontal cortex to hold the DMN down. The task-relevant signal that would normally trigger suppression is absent. So the DMN, encountering an environment that doesn't instruct it to be quiet, runs longer than it might in the familiar workspace, with less competition from the task-positive networks, with a looseness that the day didn't permit. What the DMN does when it runs freely is not entirely understood, and I want to be honest about that rather than packaging it into a neater story than the evidence supports. What seems likely, based on a reasonable accumulation of research, is that some of what the DMN does during mind wandering is useful. Memory consolidation adjacent processes, the quiet rehearsal and reorganization of recent experience. Future simulation, running low-fidelity models of upcoming situations, checking them against known patterns, updating predictions. Social processing, working through the texture of recent interactions in ways that help the social brain calibrate its ongoing models of other people. None of this is deliberate. None of it is accessible to conscious direction. It happens in the background of the wandering mind, underneath the surface content of whatever thought seems to be occurring. The mind that appears to be doing nothing is often doing something the conscious mind can't see. There's a wrinkle here that's worth mentioning, because it complicates the picture in an honest way. Excessive DMN activity, particularly the ruminative kind where the self-referential processing becomes repetitive and negatively valenced, is associated with certain mental health conditions and with the experience of worry and rumination more generally. The DMN running freely is not always pleasant. It is not always productive. A mind left entirely to itself, without any environmental engagement or soft attentional anchor, can turn its simulation capacity toward distressing future scenarios or painful past ones with the same facility it brings to more neutral wandering. The environment matters here too. A natural or low-demand environment that provides soft, involuntary attention, something for the perceptual system to engage with loosely, without directed effort, seems to allow the DMN to run without tipping into the ruminative pattern. The gentle engagement provides just enough anchor. The wandering mind has something at its periphery, something that keeps it loosely tethered to the present moment while still releasing it from task demands. A room with a window, the specific quality of evening light, the ambient acoustic texture of a space that isn't demanding anything. The DMN runs, but with a soft hand on its shoulder. By nighttime, something is different in how the DMN operates. Sleep pressure, the accumulated adenosine, the rising homeostatic drive, begins to affect the quality of waking thought in ways that overlap with some DMN states. The boundary between directed and undirected thinking becomes less distinct. Thoughts arrive with less editorial control. Images appear without being summoned. The wandering becomes looser, less narratively coherent, the simulation engine running on lower power and producing stranger outputs. This is not the DMN malfunctioning, this is the DMN approaching sleep. The network that runs when the mind is with itself begins to produce the kind of uncurated, associative, image-rich content that transitions, eventually, into dreaming. The boundary isn't sharp. The DMN that was running during the quiet evening, when the environment stopped asking for task performance, is the same network that will be running, in altered form with altered inputs, when sleep begins. The leash was already off. Sleep just takes it the rest of the way. Right now in this room, with this particular quality of dark and quiet, the DMN is doing what it does when the task demands of the day have released their hold. It's moving through something. You might notice it as thoughts that don't quite connect, fragments, a face from earlier, a question without a question mark. The sensation of being on the edge of something you can't name. That's not restlessness. That's the network doing its work. Let it. The nervous system doing this right now is old. Not in the way that feels meaningful when someone says it, not old as a rhetorical gesture toward depth or wisdom, but old in a way that has actual structural consequences. The hippocampus encoding your location tonight, the locus coeruleus that stirred when the environment changed, the olfactory pathways that bypass the thalamus to reach the amygdala directly. These are not recent developments. They are not innovations that arrived with language or tool use, or any of the elaborations that distinguish the human brain from its predecessors. They are ancient. The hippocampus, in recognizable form, appears across vertebrates so divergent from each other that their common ancestor lived before the dinosaurs. Fish have it, reptiles have it, birds have it. Its basic architecture, the layered cortical structure, the place cell populations, the connections to the entorhinal cortex, has been conserved across hundreds of millions of years of evolution, with a fidelity that suggests it solved something important early and kept solving it. What it solved was, roughly, where am I, what happened here, and should I return? This is easy to state and hard to fully absorb. The brain sitting behind your eyes right now contains structures that were already doing spatial encoding in the ancestors of every vertebrate alive today. The place cells firing in your hippocampus as you lie in this room are running a program that was already running, in simpler form, in smaller brains, in creatures that had no cortex to speak of, when the continents were arranged differently than they are now. The program has been modified, elaborated, integrated with newer structures in ways that produce the particular complexity of human spatial cognition. But the core of it was not invented for human needs. It was inherited, preserved because it kept working, because the animals that had it survived more reliably than those that didn't, and survival was the only filter. For most of the time this system has existed, the environment it operated in changed constantly. Not by the day, the way modern environments change, a different screen, a different room configuration, a different social context, but by season, by territory, by the slow grinding pressure of resource availability and predation, and the need to navigate landscapes that didn't stay the same. The brain that could track context, that could map not just space but the history of a space, the dangers that had been associated with it, the resources that had been found in it, the specific sensory signature that made it identifiable. That brain had a meaningful advantage. The brain that couldn't distinguish between a familiar safe territory and an unfamiliar one, that treated all spaces as equivalent, that failed to update its behavior based on environmental context, operated at a consistent disadvantage. Context sensitivity wasn't a feature added to the nervous system. It was a prerequisite for survival. The locus coeruleus and its norepinephrine system, the structure described several chapters ago, the one that stirs when novelty arrives, is old in the same way. A small, dense cluster of neurons with projections reaching nearly everywhere, responding to environmental change with a chemical signal that increases neural acuity, that sharpens the processing of new information, that may strengthen the encoding of new spatial maps. This response is present in fish, in amphibians, in reptiles, in every vertebrate lineage that has been studied with enough resolution to see it. It predates the neocortex by an enormous margin. It predates language, culture, agriculture, and every organizational structure human beings have built on top of the biological foundation. It was responding to new environments, sharpening attention, increasing encoding fidelity, long before there was anything like a human being to benefit from it. When you walk into a new room and feel briefly more alert, when the edges of things seem slightly clearer, and the sounds carry slightly further, and the air seems to arrive with slightly more information than air normally does, that is not a uniquely human experience. It is a vertebrate experience, hundreds of millions of years old, running in your brainstem right now because it ran in the brainstem of every ancestor who survived long enough to reproduce. There is something in the olfactory system worth returning to here, in this context. The directness of the olfactory pathway, The fact that smell reaches the amygdala and hippocampus without thalamic relay, bypassing the sensory processing bottleneck that every other sense passes through, is not an accident of anatomy. It reflects the evolutionary priority of olfactory information in the ancestors of modern vertebrates. For much of vertebrate evolutionary history, smell was the primary sense. Vision for early vertebrates operating in dim or murky environments was limited in range and resolution. Hearing was retinal but less elaborated than in later lineages. Smell was the sense that detected predators before they were visible, that tracked food sources over the distance, that identified territory, recognized individuals, assessed reproductive state, evaluated the chemical history of a location in ways that no other sense could approximate. The brain that processed smell slowly, that routed it through additional relay stations before allowing it to influence behavior, was the brain that responded too late. The direct pathway is not primitive in the pejorative sense. It is efficient in the most fundamental sense. It was the right architecture for the problems olfaction was solving, and it has been preserved because those problems haven't disappeared, only been layered over by newer ones. The default mode network, that collection of midline regions that activates during self-referential thought, during simulation, during the mind's wandering war, has its own evolutionary story, though it is harder to trace with the same clarity because the structures involved are more recently elaborated and less conserved across distant lineages. What can be said is that the capacity for mental simulation, for running internal models of possible futures, for rehearsing social scenarios, for considering what another individual might be thinking, appears to have been strongly selected for in social primates. The brain that could model other minds, that could simulate outcomes before committing to them, that could run a kind of internal prediction engine for social and environmental situations, navigated the complexities of group living more successfully. The DMN, or something functionally like it, may represent the neural substrate of that capacity. When it runs at night, in the quiet after the day's task demands have released, running its associative, image-rich, loosely narrative content through the regions involved in self and memory and social cognition, it may be doing something that served our ancestors in ways that are no longer visible to us, something that survival required often enough that the system was built to do it automatically, in the gaps between demands, whenever the environment permitted. The mind that wanders at night has always wandered at night, long before there were words for what it was doing. What the modern environment does to all of this is worth noting briefly, without making it into more than it is. The systems described across these chapters, context-sensitive, novelty-responsive, spatially precise, evolutionarily ancient, were calibrated for environments that changed at the pace of seasons and migrations and territorial shifts. They are running now in environments that change at the pace of screens and notifications, and the particular modern pressure of a day that never quite stops producing demands. The mismatch is real. The chronic load discussed in the previous chapter is partly a mismatch artifact, a stress response system calibrated for discrete acute threats, running continuously against a background of diffuse persistent ones. The attentional depletion from Chapter 5 is partly a mismatch artifact, an inhibitory system evolved for intermittent focused demands, running under conditions of near-continuous directed attention. None of this is catastrophic. The systems adapt imperfectly in the ways biological systems always adapt, through the mechanisms available to them, which are the mechanisms that were available to their ancestors, which weren't built for this but are doing their best with it. The ancient architecture is running in a new building. It recognizes some things, doesn't recognize others, keeps doing what it was built to do regardless. Tonight, in this room, the systems are doing what they've always done. The hippocampus is holding the map. The locus coeruleus is quieted, finding nothing new left to report. The olfactory pathway confirmed in the first breath that this air is known. The default mode network is running its loose evening content through structures that were running similar content long before the species was what it is now. The context is recognized. The ancient systems agree this place is known. This darkness is familiar. The body that is here has been here before, and the tissue remembers it. and the memory goes deeper than any memory you could put into words. It goes all the way down, into the architecture itself. You probably didn't notice it happening. That's the thing about this particular kind of recovery. It doesn't announce itself. It doesn't arrive with the clean relief of sleep or the obvious exhale of finishing something difficult. It moves through the body without asking for attention, which is partly why it can move through the body at all. It happened, maybe, on a walk that achieved nothing, or sitting somewhere with a view of something that moved, trees in mild wind, water doing what water does, the particular slow drift of clouds that the eye follows without deciding to. You weren't trying to rest, you weren't trying to recover, you were just there, and the body, finding itself in conditions that didn't demand very much of it, quietly began doing something it couldn't do while the demands were running. Rachel and Stephen Kaplan spent years studying what happens to people in natural environments, and what they found, through experiments that were simpler than the conclusions they produced, was that something measurable changes in people after time in low-demand outdoor settings, and it changes in the direction of restored capacity rather than depleted capacity. Attention tasks performed after time in nature were completed more accurately. Sustained focus lasted longer. Irritability, which is one of the more reliable behavioral markers of attentional depletion, was reduced. The cognitive resources that had been running low came back to something closer to their morning levels. Not because the people slept, not because anything difficult was resolved, because the environment stopped requiring the kind of processing that depletes, and the body used the gap. The gap is the important part. When directed attention is running, when the prefrontal inhibitory mechanisms are active, when the brain is filtering distraction and maintaining task representations and sustaining focus against the pull of everything that isn't the task, a specific metabolic cost is accumulating. Not dramatically, not in a way that registers as exhaustion in real time, but accumulating. What restores it is not simply rest in the passive sense, lying still in a demanding environment, a bright, noisy room, a space associated with work, a context that keeps generating task-relevant signals, doesn't produce the same restoration as the same amount of time in a low-demand, softly engaging environment. The body needs the environment to cooperate. It needs the context to stop asking. Involuntary attention is the mechanism, the kind that doesn't require effort, captured by the environment rather than deployed toward it. Moving water holds it. Peripheral complexity in natural settings holds it. The unpredictable but non-threatening motion at the edges of an outdoor space holds it. These things engage the perceptual system just enough that the brain isn't left entirely without input, but not so much that the directed attention system needs to activate to process them. The directed system gets to stop. The inhibitory effort that has been running since morning and the constant active suppression of distraction, the sustained maintenance of focus, releases, not dramatically, not all at once. The way a hand releases something after holding it for a long time, in stages, the tension leaving in layers, the grip loosening before the thing is fully set down. During that release, something else runs. The prefrontal cortex, briefly freed from its role as attentional gatekeeper, shifts its activity. The default mode network, which spent the day being suppressed, is allowed to run. The body's autonomic balance tilts, slightly, measurably, toward parasympathetic dominance. Heart rate variability, which is a reasonably sensitive index of autonomic state, increases. Cortisol, if it was elevated, begins a descent it couldn't quite manage while the demands were maintaining it. None of this is dramatic, none of it feels like anything in particular while it's happening. This is the part I keep returning to, because it seems important and also slightly strange. The restoration is not perceptible in real time. You can't feel it occurring. You can't monitor it the way you can monitor, say, the cooling of a hot drink or the slow easing of a sore muscle under heat. It's happening below any level of sensation you have access to. The autonomic shift, the cortisol descent, the prefrontal release, these are measurable in a laboratory, with instruments sensitive enough to detect what the body is doing without the body announcing it. But to the person in the restorative environment, there's often just not much. A vague absence of urgency. A mild quality of not quite boredom that doesn't tip into restlessness. The detection usually comes later. The walk ends. You return to the demanding environment. And somewhere in the first few minutes of re-engagement, you find, without expecting to find it, that things are slightly easier than they were before. The focus holds a little longer. The irritability is a notch lower. The task that felt stubborn in the morning offers slightly less resistance now. You find out the recovery happened by what it left behind. There's a study design that keeps appearing in this literature in various forms that I find quietly clarifying. Participants are given an attentional task, something that taxes directed focus, usually sustained and slightly tedious, until their performance starts to degrade. Then they're split. One group goes for a walk in a natural setting, one group goes for a walk in an urban setting, one group rests quietly. When all three groups return and take the same attentional task again, the nature walk group consistently outperforms the urban walk group and often outperforms the rest group as well. The urban walk didn't restore, it continued depleting. Different demands, same attentional cost. Traffic, signage, navigation, the social processing required by crowds. These things engage directed attention even when the person isn't aware of deploying it. Rest without environmental cooperation doesn't fully restore either. The environment has to participate. What the body is not doing in these moments is also part of it. Not processing threat signals, not maintaining social vigilance, not tracking task deadlines or unfinished items or the ambient pressure of incomplete demands. Not running the low-level cortisol elevation that sustained pressure requires. Not holding the muscles of the jaw and neck in the faint persistent readiness that demanding contexts install without asking. The list of what stops is longer than the list of what starts. Recovery is, in large part, a set of things ceasing. This room right now is doing something similar. Not identical to a walk in a natural setting, the mechanisms overlap but the specifics differ. But the basic condition is the same, an environment that is not asking for directed attention, that is not generating task-relevant signals, that the body has learned over many nights to associate with the cessation of demands. The muscles that have been slightly tensed are somewhere in the process of releasing. The cortisol that remained elevated past its natural evening descent is finding its way toward its nadir. The prefrontal cortex is gradually withdrawing from its role as gatekeeper. You didn't try to make this happen. You don't need to try. The body knows what this room means. It started recovering when you lay down. It's still recovering now. Novelty has a half-life. The first night in any new space is the most neurologically eventful, the locus coeruleus most active, the place cell ensemble assembling from scratch, the olfactory system receiving unhabituated input, the auditory cortex still processing the room's sounds without prediction. Every system engaged at a level it will never quite reach in this room again. By the second night, something has already changed. Not dramatically, not in any way you'd notice or name, but the place cell ensemble that fired on night one has been partially consolidated during the intervening sleep, the hippocampal replay that happens during slow-wave sleep, strengthening the synaptic connections that hold the spatial map, making the patterns slightly more stable, slightly faster to reinstate on re-entry. The sounds that kept the auditory cortex alert on night one have been partially habituated to. The olfactory system has already begun reducing its response to the room's particular atmosphere. The second night is quieter, neurologically, than the first. The third quieter than the second. This is how a room is learned, not through any deliberate act of memorization, not through conscious attention or intentional study of the space, through repeated exposure, the same sensory input arriving again and again, each arrival allowing the brain's predictive model of the room to refine itself slightly, to close the gap between what the room produces and what the brain expects it to produce. The gap is the important variable. On night one, the gap is large. The brain has no reliable model of this room, So the incoming sensory information, the temperature, the acoustic texture, the visual layout in peripheral vision, the particular quality of the darkness, all of it arrives with high prediction error. The brain is processing it fully, building the model from scratch, assigning it to the new place cell ensemble that is being assembled around it. On night 30, the gap is nearly closed. The brain's model of this room is so accurate that most of the incoming sensory information simply confirms what was already predicted. The prediction error is small, and small prediction error is, in the language of the nervous system, a successful model. The room has been learned. The brain knows what it will find here before it finds it. What this process looks like at the cellular level is a particular kind of synaptic change called long-term potentiation, a strengthening of the connections between neurons that have fired together repeatedly, a physical alteration in the efficiency of the synapses that hold the room's representation together. The phrase HEB is associated with neurons that fire together wire together is a simplification, but a useful one. Every time the place cell ensemble for this room activates, the synaptic connections within that ensemble become marginally more efficient. The pattern becomes marginally more stable. The threshold for reinstating the full ensemble from a partial cue, from a fragment of the room's sensory signature, drops slightly, until eventually the threshold is very low. A single smell, the particular quality of this darkness, the acoustic texture of this specific quiet, any one of these is enough to begin reinstating the full ensemble. The room is known so completely that almost any fragment of it is sufficient to reconstruct the whole. This is what familiarity is, in neural terms. Not a feeling exactly, though it produces feelings. A model that has been refined through repetition until its prediction error is low, held in synaptic connections that have been physically strengthened by a repeated co-activation, accessible from almost any fragment of the original encoding conditions. The comfort of a familiar room is partly this: the nervous system encountering an environment whose sensory output it can predict with high accuracy, which means the processing is efficient, the prediction error is small, the locus coeruleus has nothing new to report, the auditory cortex has nothing unfamiliar to resolve. The system is running smoothly, low metabolic cost, high predictive confidence. What the body experiences as comfort is, underneath, the nervous system doing its job without friction. The extended metaphor I want to use here and then let go of is of a path through grass. The first time you walk a route through long grass, you make your own way, pushing through, finding the path as you go, each step uncertain enough to require active navigation. By the 10th time, the grass has begun to flatten where you've been walking. By the 30th time, a path is visible. By the 100th time, the path is worn enough that following it requires almost no navigation at all. The route is physically encoded in the landscape, the way forward obvious without deliberation. The synaptic changes in the hippocampus are something like this. Each exposure to the room is a pass through the grass. Each night's sleep, with its hippocampal replay, its slow wave consolidation, its transfer of spatial information from temporary hippocampal storage toward more stable cortical representation, presses the path a little deeper. The place cell ensemble becomes the worn path. Reinstatement becomes, eventually, as effortless as following a route that has been walked so many times the feet find it without consulting the mind. And then the metaphor reaches its limit, as metaphors do, and what remains is just the biology. Synaptic weights. Prediction error. A model refined across nights until it knows the room the way tissue knows a groove it has always rested in. There is something worth noting about what gets lost in this process, alongside what gets gained. The alertness of the first night, the locus coeruleus active, the senses admitting more signal, the encoding operating with the enhanced fidelity that novelty produces, that is gone by the time the room is fully learned. The richness of new experience, the particular vividness that comes from encountering something without prediction attached to it, doesn't survive familiarity. It can't. It is by definition a property of novelty. What replaces it is not better or worse. It is efficient, stable, low cost. The brain, allocating minimal resources to a room it has already fully modelled, freeing those resources for other processing. The room becomes, in a sense, invisible, not missing, just no longer requiring attention, no longer generating prediction error, no longer asking the nervous system to do anything that it hasn't already done many times before. This is the biology of home, in whatever form home takes, not a feeling primarily, a state of prediction. The room you're in right now is somewhere on this curve. If you've been sleeping here for years, the model is deeply consolidated, the place cell ensemble stable, the synaptic weights well established, the prediction error near zero. The room is known in the most complete sense the nervous system uses that word. Every sensory input it produces has been expected. Every sound has a prediction. Every quality of the darkness has been modelled and confirmed enough times that it arrives without surprise. If you're newer to this space, the consolidation is still in progress, the path being worn, the model still closing its gap with the room's actual sensory output, some sounds still arriving with slightly more prediction error than others. Either way, the process is running. Every night adds something to it. Every sleep consolidates what the day's exposure began. And at some point, without announcement, without any moment that feels like arrival, the room will be fully known. The model complete, the path worn through. You've stood in a doorway and forgotten why you crossed it. Not often, maybe, but enough times that the experience is familiar. The entry into a room with a clear purpose that dissolved somewhere between there and here, leaving you standing in the new space holding the shape of an intention without its contents. This is not a memory failure, it's a feature. The brain does not experience time as a continuous flow. This is one of those things that sounds wrong at first because the subjective experience of time feels continuous. One moment following the next in an unbroken stream. Events connected to each other by the thread of awareness that runs through all of them. But what the brain is actually doing underneath that felt continuity is something more segmented. It is constantly parsing, dividing the ongoing stream of experience into discrete chunks, episodes in the language the research uses, bounded by moments that the brain identifies as transition points, a conversation ending, a task completing, a scene changing, a location shifting. The boundaries between episodes are not arbitrary. The brain looks for them at moments of significant change, moments when the sensory environment or the internal state or the ongoing action sequence undergoes a shift large enough to register as a discontinuity. A doorway is one of those moments. The research here comes largely from the work of Gabriel Radvansky and colleagues who spent years studying what they called event boundary, the moments where the brain segments continuous experience into distinct episodes, and found something specific about physical thresholds. Crossing from one room to another produced a reliable decrease in the accessibility of information from the previous room. Not just because the context changed, though context-dependent memory, as discussed earlier, contributes, but because the physical threshold itself seems to function as an event boundary signal. The brain, encountering a doorway, updates its episodic model. The previous episode is filed, a new episode begins, and the filing is briefly quite thorough. What was in active working memory, the contents of the current episode, the task being pursued, the intention that prompted the movement, gets deprioritized as the new episode opens, not erased. Deprioritized. The hippocampus, which indexes episodic memories and manages the transition between them, is busy opening a new file. The old contents slip. Working memory is limited in a way that matters here. It can hold a small amount of information in an active, immediately accessible state. 4 items, roughly, is the estimate that has held up reasonably well across decades of research, though the exact number varies depending on the nature of the items and how they're measured. The contents of working memory at any given moment are whatever the current episode has made relevant. When an event boundary is crossed, the current episode ends and the new episode begins with its own relevant structure. The previous episode's contents don't automatically transfer. If the intention that prompted the threshold crossing was represented in working memory, if it was one of the 4 or so items being actively held, the transition to the new episode creates a moment of competition. New sensory information from the new room is arriving. The hippocampus is indexing the new context. The place cell ensemble is remapping. Into this moment of transition, the old intention has to fight for its place. Sometimes it wins, sometimes it doesn't. And you stand in the kitchen, certain you came here for something, with only the certain feeling and not the something. What's happening in the fraction of a second at the threshold is neurologically dense, in a way that the felt experience doesn't reflect. The hippocampus is executing a context update, recognizing the shift in spatial context, beginning to reinstate the place cell ensemble for the new room, flagging the transition as an event boundary for episodic indexing. The entorhinal cortex is updating its grid cell representation. The locus coeruleus, if the new room is sufficiently different from the previous one, is nudging its norepinephrine output slightly upward. The olfactory system is taking its first unhabituated read of the new air. All of this in a moment that feels, from the inside, like simply walking through a door. The compression is remarkable, a fraction of a second. The nervous system executing a coordinated update across multiple structures simultaneously, each one doing its specific job in response to the same transition signal. And then, it's done. And you're in the new room, and the new episode has begun. This is why changing your environment works at the scale of mood and cognitive state as well as the scale of memory. The event boundary doesn't just file the previous episode's contents. It files the previous episode's associated state, the emotional texture, the cognitive set, the particular configuration of concerns and preoccupations that the previous context was maintaining. Not permanently, not completely, but the accessibility of that state decreases at the threshold, in the same way that the accessibility of working memory contents decreases. You walk into a different room and the worry that was running in the previous room is still encoded, still accessible if you deliberately reach for it, but not foregrounded in the same way. The new episode opens without it automatically included. The threshold resets the active contents. There is something that the research points toward but doesn't fully explain, the felt quality of crossing certain thresholds that produces something beyond the simple cognitive reset. Not all doorways feel the same. Moving from a demanding room into a restorative one doesn't just trigger an event boundary, it triggers an event boundary in the direction of a context the nervous system has already learned to associate with reduced demand, with the releasing of attentional posture, with permission to stop. The threshold in that case is doing 2 things simultaneously, closing one episode, and opening one the body already knows how to be in. The doorway you cross tonight, into this room, into the dark, into the particular arrangement of familiar sensory information that the hippocampus has been consolidating across many nights. That threshold was one of these. The episode that preceded it filed. The new episode opened. And what this episode is associated with, what the body has learned to expect when it crosses into this specific context, is the gradual release of everything the previous episodes required. The threshold was small, a few steps, a shift in the air, a change in the quality of light. But the nervous system doesn't measure thresholds by their size, it measures them by what they separate. And this one separates the day from whatever comes after it, which is this, which is already here. The hippocampus has been recording all day, not selectively, not only the moments you'd have chosen to remember, continuously in the way a system built for survival records rather than a system built for meaning. Every room you moved through today left something in it. Every threshold crossed, every context shift, every moment of novelty that stirred the locus coeruleus and sharpened the encoding. All of it is held somewhere in the hippocampal tissue, in the temporary form that new memories take before they've been consolidated into anything more stable. The hippocampus is not built for long-term storage. It is built for rapid acquisition, for taking in new information quickly, holding it in a form that can be accessed in the short term, and then transferring it during sleep. to the cortex where it can be held more permanently and integrated with everything already known. The transfer happens tonight. In the first hours of sleep, as the brain descends into slow-wave sleep, the deep, high-amplitude, low-frequency stage that dominates the early part of the night, something specific begins in the hippocampus. Replay, the place cell sequences that fired during the day's navigation, the ensemble for the kitchen, the ensemble for the hallway, the ensemble for this room, begin replaying in compressed form. Not in real time, not at the pace they fired during waking experience, but faster, the sequences running through in fractions of a second, the hippocampus cycling through the spatial record of the day at a speed that would be meaningless to conscious experience, but is precisely the speed the consolidation process requires. This was first observed in rats, in experiments where electrodes recorded from hippocampal place cells during both waking navigation and subsequent sleep. The sequences of cell firing that occurred while the rat moved through a maze appeared again during slow-wave sleep, in the same order, recognizably the same pattern, but compressed. The hippocampus was replaying the day's spatial experience. The same process runs in the human hippocampus. The rooms you moved through today are being replayed right now, or will be, in the first deep sleep of the night. The spatial sequences firing again in compressed form. The patterns that encode each room strengthening their synaptic connections through the repetition. The temporary hippocampal representation becoming gradually more stable. What the replay is doing mechanistically is something like this. During waking encoding, new memories are held in the hippocampus in a form that is relatively fragile, dependent on the continued activity of the neurons that encoded them, vulnerable to interference from new incoming information not yet integrated with the broader cortical networks that hold established knowledge. The synaptic connections that hold the new memory are there, but they are recent and relatively weak. During slow-wave sleep, the hippocampus is relatively isolated from the sensory input that was driving it all day. The thalamic gates that relay sensory information to the cortex are partly closed. The brain is, in a real sense, turned inward. And in this inward-turned state, the hippocampus replays, driving activity in the cortical regions that were coactive during the original encoding, strengthening the connections between the hippocampal representation and the cortical networks it's connected to, gradually shifting the memory toward a form that is less dependent on the hippocampus itself. This is called systems consolidation, and it takes place across many nights, not just one. A single night of sleep begins the process. The memory is not fully transferred by the morning, but it is more stable than it was at sleep onset, more resistant to interference, more deeply woven into the cortical fabric. The spatial memories encoded today, the place cell ensembles, the grid cell coordinates, the contextual details threaded through each room's representation are being woven in tonight. And because context-dependent memory encodes not just space, but the emotional and cognitive texture of each space, the load that a particular room carried, the associations built up over repeated visits, the particular quality of each threshold crossing, that texture is being consolidated too. Not as explicit memory, not as a narrative you'll be able to retrieve and recount, as something more implicit, a refined model of each context, held in the connections between hippocampal and cortical networks that will influence tomorrow's experience of these spaces without ever surfacing as a conscious memory. The brain waking tomorrow will know this room slightly better than the brain that fell asleep tonight. Not as knowledge it can articulate, as prediction, REM sleep, which dominates the later hours of the night after the slow-wave cycles have done their early consolidation work, contributes something different. During REM, the hippocampus is active in a different pattern, not the sharp-wave ripples of slow-wave replay, but a more chaotic, associative activation that seems to reflect something like recombination rather than pure consolidation. Memories are not simply strengthened during REM, they are, in some sense, mixed, brought into contact with each other, with established memories, with emotional associations, in combinations that don't follow the logic of waking sequential thought. The environmental memories from today may be connected, during REM, with memories of other environments, similar spaces, similar thresholds, similar felt experiences of moving between contexts. The brain appears to be looking for patterns across its stored experience, building associative networks that link new information to old in ways that will allow generalization. The ability to apply what was learned in one context to a novel but similar one. This is why sometimes a problem that felt intractable before sleep has a different quality after it. Not because the sleep invented a solution, but because the REM processing found connections between the problem and existing knowledge that weren't accessible in the rigid sequential logic of the waking mind. The environmental record of today is being integrated, tonight, into a web of related experience that extends back through every similar day that has been slept on. There is something I find quietly remarkable about the directionality of all this, and I want to name it carefully without overstating it. The brain is not just filing the day's experience during sleep. It is updating its predictive model of the world. Everything the previous chapters described, the place cell ensembles built in each room, the habituation to each room's sensory signature, the context-dependent encoding of the load each space carried, the event boundaries at each threshold, the norepinephrine sharpened encoding that novelty produced. All of it feeds into the model the sleeping brain is refining. Tomorrow, the brain that wakes will generate predictions for each familiar environment with slightly higher accuracy than the brain that fell asleep tonight. The model will be marginally more refined. The prediction error marginally smaller. The room slightly better known at the cellular level, the context slightly more accurately anticipated, the threshold slightly more efficiently processed. This is not dramatic. It is incremental, reliable, and continuous across every night of every life. The brain building, night by night, its model of the inhabited world. Tonight's sleep will do what every night's sleep does. It will take what the day produced, the new ensembles and the refined ones, the habituated sounds and the unresolved novelties, the context learned a little more deeply and the thresholds crossed and filed, and it will work with it, not quickly, not in any single stage. Across the architecture of the night, in the slow wave cycles that consolidate and the REM cycles that integrate, through the hippocampal replay that runs while you are entirely elsewhere, in whatever experience sleep produces, the rooms you move through today are in there, being processed, being woven into the model of a world the brain is always, quietly trying to know more accurately. By morning it will know it a little better, you won't notice. But the cells will have changed, and the next time you cross a familiar threshold, or stand in a room the body has learned, or breathe the first unhabituated breath of a new space, the model doing all of that will be tonight's model, built partly from today, refined, without ceremony in the dark. The body you have right now is not the same body that woke up this morning, not dramatically, not in any way that would show in a mirror, or register in a blood panel, or announce itself as change. But the calibration has shifted. Across the hours between then and now, the accumulated record of every space you move through, every room, every threshold, every moment of context shift, has made small adjustments to almost every system involved in how the brain models the world. The place cell ensembles are slightly more consolidated. The predictive models for each familiar environment are marginally more refined. The habituation to certain sounds has deepened. The norepinephrine system has risen and quieted in response to whatever novelty the day produced, and the encoding it sharpened is now held in synaptic connections that are fractionally stronger than they were this morning. None of this was noticed while it was happening. It rarely is. Horizontal is a biological signal. The body lying down is not simply the body at rest. It is the body in a posture that the nervous system reads as a specific state, distinct from sitting, distinct from standing, carrying its own autonomic implications. Blood pressure drops slightly as the cardiovascular system adjusts to the new relationship between heart and periphery. The vestibular system, which has been tracking orientation and movement all day, receives input that it registers as stable, as still, as no longer requiring the continuous postural adjustments that upright waking life demands. The brainstem structures that regulate sleep, the ventrolateral preoptic nucleus, which releases GABA and galanin to inhibit the arousal systems, the suprachiasmatic nucleus, which has been tracking the light cycle all day and translating it into melatonin timing, receive the signal of horizontal stillness as one more piece of evidence that the conditions for sleep are assembling. Not a trigger, a contribution. One more thing the body needed to be before sleep could begin. The room around you is doing what familiar rooms do at this hour. Its sounds are predicted. Its particular quality of darkness has been modelled across many nights and arrives now without prediction error, without novelty, without the locus coeruleus finding anything new to report. The air carries the chemical signature that the olfactory system has so thoroughly habituated to that the molecules arrive at the epithelium and produce almost nothing. No signal worth forwarding, no pattern worth assembling into conscious perception. The room is neurologically very quiet, not because it is physically quiet, though it may be that too, because the brain has learned it so completely that almost nothing it produces constitutes new information. There is a specific quality to lying still in a room the body knows well. It's hard to name because it operates below the level of language, below the level of anything that could be described as a thought or a feeling in the usual sense. Something in the nervous system recognizes the combination. Horizontal, familiar, dark, the particular acoustic and olfactory signature of this specific space. The recognition isn't conscious. It runs in the structures that learned this room over many nights of repeated exposure, in the place cell ensemble that reinstates without effort, in the cortisol rhythm that has learned to descend here, in the muscles that have learned over time that this arrangement of the body in this space means the demands are finished. The recognition produces a state, not sleep yet, the state just before sleep which has its own biology, hypnagogia, the threshold condition, the loosening of the editorial controls that waking consciousness maintains over the content of thought. Images arriving without being summoned, sounds that aren't in the room, the edges of the day's experience beginning to soften and recombine in the way that REM will later do more thoroughly. The brain already beginning to do at the edges of waking what sleep will do more completely in the hours ahead. You move through spaces today, some of them familiar enough that the body barely registered them as events. The place cells reinstating instantly, the predictive model confirming itself with minimal processing cost. The context recognized so quickly that the transition was nearly invisible. Some of them slightly less familiar, requiring a few seconds of additional encoding before the model caught up to the room. Each one left something. Not as memory in the narrative sense, not as a story you could tell, a sequence you could retrieve and examine, as calibration, as a small adjustment in the accuracy of the brain's model of the inhabited world, a fractional refinement in the prediction, a synaptic weight marginally shifted in the direction of greater certainty about what to expect from these spaces. The day was, among other things, a series of biological adjustments. They happened quietly, continuously, below the threshold of notice. And they're in the tissue now. The geography of today is already inside you. Not as images, not as anything that could be called recollection. As the particular state of the nervous system right now, shaped by every environment it passed through, carrying in its calibrated predictions and habituated responses and consolidated place cell patterns, the record of a day spent moving through rooms. Tomorrow's brain will navigate these spaces with the benefit of tonight's consolidation. It will cross familiar thresholds with fractionally lower prediction error. It will reinstate familiar place cell ensembles fractionally faster. It will habituate to familiar sounds fractionally more smoothly, the prediction already in place before the sound fully arrives. The body learns the world in increments, each day providing the material, each night doing something with it. Right now the material is today's. The rooms are in the hippocampus, still in their temporary form, waiting for the slow wave sleep that will begin the consolidation. The contextual details are threaded through the spatial records, held together in the fragile early state that new memories occupy before sleep makes them more permanent. The thresholds are indexed, the event boundaries are filed. The sensory signatures of each familiar space are woven into the existing models, which are already slightly better than they were this morning. All of this is waiting for what is already beginning. The breath is slower now than it was an hour ago, not because you tried to slow it, because the body horizontal in a familiar dark room with the demands of the day behind a threshold that was crossed some time ago defaults to this, the respiratory rate dropping, the tidal volume adjusting, the autonomic balance tilting toward the parasympathetic without being asked. The jaw, if you notice it now, is looser than it was. The muscles along the neck. The particular holding that the day installs, so gradually that it never announces itself, is somewhere in the process of releasing. Not all at once, not dramatically, but in the quiet, incremental way that biological processes release when the conditions that maintain them are no longer present. The conditions are no longer present. The room is familiar. The body knows it. The day's geography is already in the tissue. And what comes next, and the long, slow processing of everything the hours produced, the hippocampal replay and the cortical weaving, and the associative integration of REM, is already beginning at the edges. You don't need to do anything for it. You just need to be here, in this room, in the dark, which the body already knows, which has already, in every way that matters biologically, been arrived at.